Thank you, Carla, for having me here. Thank you to President and Mrs. Ayoun for hosting us. And hey, Skip, it's good to see you. Good to be on a platform with you again. And I know the last time you heard me speak, I kind of blew your mind. With talking about 5 sexes. So today I'm going to talk about tomato soup. And you all have the handout here. I think Carla's introduction was really perfect when she said that what she wanted to do was to give us agency to talk about, to think about all these extraordinary things that are going on in the biomedical sciences and beyond, and the ways in which DNA and ideas about genetics are just permeating our lives at many different levels. So I thought to give us agency, I wanted to start with some basic definitions, and I thought rather than tell you about embryos, it would be easier to tell you about tomato soup. So the first thing I want to say, and this is the slide 2 on the top of your handout, is that DNA is not the same thing, and that's supposed to be an equal sign with a blue slash through it, does not equal the biology of a developed organism. A developed organism, any of the adults in this room, even very little children, are a great deal more than their DNA, and their biology represents a great deal more than their DNA. So if we ask, though, what does DNA offer us, I would say that the DNA sequence, that series of bases that genome sequencers now are measuring and the order in which they appear, could be equated to an ingredients list. In that sense, we all have somewhat different ingredients unless we're identical twins. What ingredients, if you're making soup, what ingredients you put in to start with do matter in terms of the ultimate flavor or production of the soup. But our biology, when I say it's not the same as our DNA, is the completed bowl of soup. And of course, the bowl of soup is never completed, and really until we die, just as a bowl of soup that you leave in your fridge continues to develop in new and colorful, often ways. Not necessarily edible, but definitely colorful. So if you really want to get into what happens during development, you can liken that to the instructions that come with a recipe. So I've included instructions for one tomato soup that says preheat the oven to 450 degrees, strain the chopped canned tomatoes. I won't read the whole recipe. So you have, when you get a recipe, you get a list of ingredients, but you also get instructions on how to assemble the ingredients. How you treat the ingredients can really matters for the final product. So in this case, I've suggested in slide 6 that there are lots of things that happen to the bases on our DNA that affect how they function in the organism, and these are generally called, biologists call them epigenetic changes, and so they're sometimes they're heritable, but they aren't the same as a mutation. That is, they're chemical modification of the DNA that can get stripped off later, never changing the basic gene sequence. But you can think of those modifications to DNA are as metaphorically similar to how we change or modify the ingredients in our tomato soup recipe, whether we roast the tomatoes or saute them or puree them. Each choice that we make modifies the ingredients and ultimately the taste of the soup. And the history of the organism matters too, or the history, and by history here I mean the order of processing and adding ingredients. So it makes a great difference to the final product, to the final biology, to the final taste of the soup, for example, whether you saute the onions at the very beginning and then add the other ingredients, or whether you make the soup and then you sprinkle raw diced onions on top. It's the same ingredients, onions, but depending on the order in which you add them and the way in which you modify through different cooking choices, you end up with a very different product. And the different choices mean, in the case of the soup, different tastes. And then last on this list, the organism's characteristics depend on when the soup is consumed. So it could be consumed as a hot soup, as a cold soup, as fresh. You could reheat it. If you reheat it, it's going to make a difference whether you heat it on the stovetop or microwave it, or whether you take it and use some kind of solar, tomato soup, solar tea kind of thing. So I think it's very important to keep these distinctions in mind as you hear people say things like what we're likely to get. That is, and I think you said that, Carla. You said, knowing our DNA sequence, it's awesome, it tells us what diseases we're likely to get. But it doesn't.
But it doesn't.
Except in very few rare cases. So it tells us what some of the ingredients are that then are going to get treated throughout the life cycle in all of these different ways to produce either a disease process or not, depending on what happens. So the take-home lessons that I want to give, and this isn't on your handout because I just wrote it today, are the initial ingredients are important. That is, the DNA, knowing the DNA sequence is important. But processing and production, I would argue, counts even more. The translating back into DNA and the question of identity, let me make a few points about health identity. We are spending hundreds of millions of dollars aimed at developing personal genomics with the promise of improved health. In the past 50 years, and even before, our biggest gains in health, however, have been from public health measures, not from knowing anything about DNA. We didn't need to know about DNA to improve the rates of heart disease. By improve, that means make them decrease, by learning the importance of controlling blood pressure, of stopping smoking, of using pharmaceuticals in various ways to help with that. We've had decreases in cancer in many different kinds of cancer, and that we've done by controlling carcinogen exposure. Again, smoking is one good example of that, keeping workers away from asbestos, another example, and through early detection, which is a public health epidemiological measure. Don't need the DNA for that. And we have looming health problems, which have exploded in the last 50 years, and the fact that they've happened so quickly means that they are not at heart because there's been natural selection for some disease process, but because something else is going on. One such example is the increase in type 2 diabetes, which is clearly not related to massive genetic changes in our populations, but are related to changes in how we make our livings. That is, we don't do physical work in the same way that we did several generations ago. Big city living, unavailability of healthy foods in many areas of big cities, and most importantly, I think, profound changes in our food supply, especially bringing us to our food supply to a huge reliance on corn sugar and starch in our prepared foods, and this massive, massive increase in sugar consumption. And I'm just going to tell you, in 20 years, in the US, we have increased our per person yearly sugar consumption from 20 pounds to 135 pounds of sugar per person per year. That's an astounding 2 to 3 pounds, and some of that's hidden in high fructose corn syrup, by the way. I mean, it's not all what we take from our sugar bowl. But so it's so it's not surprising, considered that highly refined sugars in the form of table sugar, corn sugar, high fructose corn sugar are being processed into so many foods, such as bread, breakfast cereal, mayonnaise, peanut butter, ketchup, spaghetti sauce, and a lot of the microwavable meals. So there is one sense, and I am fascinated by the DNA analysis, and it's telling us amazing things about developmental processes. So I'm not saying no, but I think the question for this group to consider is where do we put our resources? If we know now how to stop the epidemic in type 2 diabetes, and it's the heart of that problem is our food supply and our relationship to physical activity, why are we not putting public health measures? We're putting some, but in those hundreds of millions of dollars, why isn't that going into public health measures rather than into DNA sequencing? And so those are choices we can make. And one doesn't have to, as I said, I really am fascinated by what we're learning about humans from the human genome and from other genomic work, a lot of which, by the way, we can actually find out by studying fruit flies, because that's one of the most amazing things that's happened in the last 30 years, is that we've learned how similar genes are in terms of their function in all sorts of animals, including humans. But to the extent that we're worrying about certain kinds of social problems, and especially health problems, I think we can talk about how we structure our priorities and whether learning more about DNA is the most important thing to do. So.